Hello again, biologists. Mr. Kinnear here with another video lesson for higher biology. We're on Unit 1, DNA and the Genome, and today we're looking at structure of the genome, which is key area 1.5.
And this is how it fits into the whole unit. We've looked at structure and organisation of DNA, its replication, gene expression and differentiation. The structure of the genome fits in here before we move on to mutations, evolution and genomics. We're going to look at the structure of the genome and what it does, but first of all, let's think about what it is. It is the sum total of all hereditary material within an organism. And that's usually the complete complement of DNA, but in some viruses it could also be RNA. We think about it like writing a story, where the words are coding sequences of DNA and spaces and other characters are non-coding sequences. And again, if you think about reading a book, if all the words were jumbled together without any punctuation, spaces or other characters, it wouldn't make much sense, and the same is true of your DNA. The genetic information is carried in linear or circular chromosomes as well as in mitochondria and chloroplasts. And the human genome consists of 3 billion nucleotides, approximately 28 to 25,000 genes, and they're arranged on 22 autosomal chromosomes and a pair of sex chromosomes, and obviously the combination being different for males and females. So in most organisms, the genome consists of genetic information coded in DNA. And the genes are the sequences of DNA bases that code for proteins, but there's also a large number of non-coding regions. In fact, in eukaryotes, only a tiny proportion, around about 2% of your genome, is actually genes that code for proteins. So what is the rest? Well, it used to be thought that the non-coding regions of DNA was just junk DNA, stuff that just didn't do anything. And to be honest, there is still a lot of that. We are carrying genetic material that's coming from viruses and bacteria through the course of millions of years in the evolution of humans. However, recently we have made more discoveries and found that non-coding sections are important for things like regulation of transcription, the transcription of non-translated RNA. There are elements of protection, and although we're not going into the detail of this today, think about the end of your shoelace. You normally have what's known as an eyeglot at the end of that stops it unravelling, and you could poke it through the hole to tie your shoelace. So that is an element of protection, much like, as you can see from this diagram or picture, there are protective ends on the chromosome that effectively stop it unravelling or being damaged or losing genes should the chromosome actually be damaged. And there is other bits of DNA which as yet we don't know what it does. So in terms of the regulation of transcription, RNA polymerase, which we know is involved in the transcription of a gene, can't initiate the process on its own. It needs some assistance from transcription factors. Some of these are called activators. They're bound to non-coding regulator sequences of DNA. So the activator binds to non-coding regulator sequences of the DNA, causing a bending of the DNA strand, bringing the regulator close to the promoter of the gene to be transcribed, then beginning the transcription process. Non-translated RNA is something we have discussed during the gene expression key area. It's not translated into a polypeptide. So for instance, tRNA, it will be coded for within your genetic code on your DNA, but it's not converted into polypeptide. Its job is to transfer amino acids during translation. The ribosomal RNA, again, forming the ribosome, not converted into a polypeptide. But there are other RNA fragments that are involved in splicing and regulation that we haven't discussed previously, but those are 3 key bits of non-translated RNA that will be coded for in your DNA code, but never make it to be a polypeptide. And that ends a very quick video lesson for today. So today we said that the genome of an organism is its hereditary information encoded in DNA, and for some viruses that is encoded in RNA. Coding regions are sections of DNA which contain genes. Much of the genome is non-coding. and those non-coding regions are involved in the regulation of transcription, producing non-translated RNA and elements of protection. So hopefully that's been helpful. If you have any problems, questions or queries, get in touch and I'll speak to you again soon. Bye.